Hello, this is talk number 19 in the series from the 7th edition of Principles of Biochemistry by Albert Lehninger. Today we'll be discussing further material regarding the role of lipids in membranes and forms of membrane transport. Today we will start with the discussion of fusion membranes, fusion proteins. And fusion proteins are those that mediate membrane combining through neurotransmitter synapses. And this is an example of that. So we talk about this process where you have cytosol with a secretory vesicle here, and neurotransmitters fill a vesicle approaching the plasma membrane. So these are the neurotransmitters in this vesicle, and you've got recognition points B snare and T snare, and the B snare and the T snare bind to each other, and they zip up from the amino terminal, and they draw the two membranes together like this. And then by the zipping causes curvature and lateral tension on the bilayers, favoring hemitransfusion between the outlier leaflets. So hemitransfusion of the outer leaflets occurs, they come into contact, then there's complete fusion that creates a fusion pore, and then the pore widens, the vesicle contents are released outside the cell. So the mediating components are the V snare and the T snare, and this is how the method by which neurotransmitter particles are released into the cell. So some elements of this are of critical import. Botulinum toxin interrupts the action of these fusion proteins and stops neural synapse transmission. You also have cadherins, selectins, and integrins. These are surface adhesion proteins which mediate transfer. The solute transport, this is represented most easily in laboratory situations by osmotic permeation of semi-permeable membranes. So when you have simple diffusion, you have a chemical gradient, a semi-permeable membrane, and that governs that. There are passive transporters, which have simple facultative movement with a concentration gradient from higher to lower. And then active transport, where substances move against a concentration and membrane potential gradient. And this, because this is a thermodynamically unproductive reaction, you have to require energy or transfer with another molecule in order to affect that type of transport to occur. The movement problem, of course, is that the molecular environment of the transport substance, you have to get from an aqueous solution through a lipid hydrophobic bilayer and then back to aqueous substance again. So that's a bit of a problem. The membrane proteins lower the activation energy, and they employ non-covalent interactions, and they are stereospecific. So the transport channel is lined by proteins, and this ameliorates the hydrophobic, thermodynamically unfavorable situation of moving this material over a lipid bilayer. There are additionally ion channels where you have transport through a gate mechanism. There's a faster rate of diffusion there, so we're talking about sodium, potassium pumps, and that process is controlled by a biological signal or equilibrium of an electrochemical gradient. We have erythrocyte glucose transport as a prime example of this. This is a model of passive transport. These obey rules of Michaelis-Menten kinetics. The glucose is equilibrated between the erythrocyte and the surrounding plasma. Glucose is transported across a helical protein channel. There's a family of GLUT or glucose transporters. They are tissue specific. So in the passive transport model, one example is a chloride bicarbonate exchange involving carbon dioxide from tissues transferred to bicarbonate, bicarbonic anhydrase in the erythrocyte. The bicarbonate re-enters plasma, and in the lung, the bicarbonate re-enters erythrocyte and is transformed to carbon dioxide and is exhaled. You get chloride bicarbonate exchange. This increases the capacity of the erythrocyte transport, and the chloride and bicarbonate ions move oppositely, and this is an electroneutral state. You can have two substances move in tandem, either oppositely or in concert, or one substance at a time. For active transport, examples of this are calcium, phosphorus. These move against gradients, and you have to utilize energy or another linking substance to affect the transport of calcium and phosphate against gradients. So the free energy change for the transport, the Gibbs energy is equivalent to the gas constant times temperature in Kelvin, natural log of C2 over C1. And the R is, again, 8.315 joules per Kelvin, and the temperature is 298 Kelvin. Now, if ions are the gradient substance, then you're looking at, instead of C2, C1, you're looking at omega, where Z is the ion charge, F is a Faraday constant, and the delta omega is the transmembrane potential in volts. The membrane potential is about 0.05 volts, with the cell interior more negative relative to the exterior, and it's generally a tenfold ion difference across these membranes. There's another category called P-type ATPases, and this is a family of active transport proteins, and this is a driving force for secondary, in other words, two substances transported in active transport, and is also the basis for neuron electrical signaling. And this is what you see with calcium, sodium, and potassium ions. There are protein pump ATPases. There are V-type and F-types found in plants, bacteria, and archaea. ABC transporters or ATP-driven transporters of multiple substances, which have a wide variety: lipids, biosalts, metal ions, amino acids, peptides, proteins, hydrophobic compounds. And usually one polypeptide has forms for two domains for nucleotides and helical channels as a part of that. There are also multi-drug transporters. A human ABC transporter is found in the placenta, blood-brain barrier, and tumor resistance. And the latter, the tumor resistance, is subject to transport inhibition. So these are additional forms that involve ATP utilization. Ion gradients, you can couple sodium and potassium transport, the sodium potassium pump. These can be coupled to other solutes, for example, lactose transport, presence in E. coli. You can couple it to proton transport and make it part of a family. There's also intestinal transport, so glucose and amino acids are coupled to sodium transport mediated by sodium potassium ATPase. And generally you can get two sodiums out and glucose out with two sodiums in and glucose in. And this balances the electrical and chemical potential given the charges that can exist in glucose at that physiologic pH, and the charges help balance all this electrostatically. So when we look at this, at the intestinal lumen, we have the apical surface of the intestine right here. This is the lumen, this is the interior, and this is the exterior. So we have the villi of the intestine, these microvilli. You can see these on the light microscope at high power. And you have two sodiums coupled to glucose. They're brought in through the sodium glucose symporter, which is driven by high extracellular sodium. So it's brought in here, and the sodium then is brought through the inner part into the basal with transport of potassium to maintain electrostatic neutrality, and these charges balance out, and you have glucose brought in. So this is the means by which glucose is brought into the cell, and this is the glucose importer, GLUT2, which facilitates the downhill efflux. So the bioenergetics of this process are rendered favorable because of the transport and maintenance of charge in the intestine. Aquaphorins facilitate water movement across membranes. These are also tissue specific. There's a low activation energy with rapid and continuous movement. This channel is a transmembrane helix. It's a narrow channel, so larger molecules are excluded and they are aligned by polar groups. These are aquaphorins for water. Ion selective channels are distinguished from ion transporters. There are three differences. One, the rate of flux is much greater than you would see in ion transporters with unrestricted diffusion. Ion channels are not saturable. There's no maximum to that. And they are often gated to a cellular event, either an associated protein that opens and closes a channel, and you can experimentally measure the ion channel in the voltage. These are model ion channel systems. The potassium channel in bacteria is a core stabilized by helices, provide a microenvironment to the correct size, and allowing potassium to move in a single file passage. So what this looks like is that there's a backbone carbonyl oxygen, which is right here, backbone carbonyl oxygen, which forms a cage, and this cage fits potassium ion precisely, replacing the waters, the hydration sphere. So the waters are excluded here, the potassium lines up single file. They're alternating potassium sites, they're occupied, blue or green, and these are stabilized by an alpha helix, which stabilizes potassium. It is brought in to the interior of the cell with the hydrating water molecules that are present within the interior of the cell. So you can bring the ion in a pure form across the membrane with the help of these stabilizing bracket molecules. What factors are critical for neural function? These are highly selective for sodium voltage gated regulated. The nicotinic acetylcholine receptor is a signal from the muscle neuron to a muscle fiber, which signals contraction. And acetylcholine is another factor. It creates an ion channel opening and passage of sodium, calcium, or potassium, causing contraction. And there's a whole family of related transport mechanisms that are grouped under acetylcholine. We'll be discussing acetylcholine in greater detail in due time. We also have biosignaling, and this is a means by which membranes convey information to cell constituents. This information is conveyed chemically via chemical transduction, and these are extremely specific interactions. So the three critical things you want with transduction with the information transfer is specificity, amplification of the signal, and modularity. In other words, multiple proteins can have multivalent affinities. Also, integration, where two signals provide opposite effects. You can have signaling molecules react in opposing means. And then in number 6, the final one, is a localized response, where the messenger acts locally and is degraded locally, and the message, the chemical message, similarly acts locally and is degraded locally. So we have 4 broad categories that we'll be discussing. G protein, these are coupled receptors. They generate a second messenger, such as epinephrine, vision, olfactory. Second model receptor enzymes, which are where the ligand binds extracellular. extracellularly. The enzyme acts intracellularly, such as tyrosine kinase, epidermal growth factor, insulin receptor. A third broad model is gated ion channels. We discussed a little bit of that. And finally, nuclear receptors, hormone ligands invoke a protein production, estrogen being a prime example. The nomenclature varies, not necessarily related to function. We have a G protein coupled receptor where the hormone binds. G protein exchanges bound GDP for free GTP from the cytosol. G protein is released, binds to a second protein, and is a second messenger released. So one example of this is a beta blocker where epinephrine binds to adrenergic receptors. Cyclic AMP is the second messenger. Agonists will bind a natural ligand, whereas an antagonist will bind and block the effect of the natural ligand. And these are involved in glycogen and fat catabolism. Adenyl cyclase is a second messenger, and that produces cyclic AMP, which activates protein kinase A, and that phosphorylates enzymes used in glycolysis. So glycolysis is accelerated and amplified using this cyclic AMP signal. It's a cascade effect. And this effect ends when you have decreased epinephrine, it's consumed, or through hydrolysis of GTP bound to G protein. Hydrolysis of cyclic AMP also stops the process. And you can also phosphorylate the receptor and activate it that way. You don't need to have the other effects of the G protein or cyclic AMP hydrolyzed. And cyclic AMP is a second messenger for numerous hormones and other signals, glucagon, corticotropin, parathyroid hormone, glucagon particularly. And these hormones inhibit cyclic AMP. They do this through activation of inhibitory G protein, and that's what stops the cyclic AMP formation. So this is what this molecule does biologically. You can also have adapter proteins. They act in concert with cyclic AMP receptor site. These affect multiple responses, and they're highly specific. So some other examples of second messengers are diacylglycerol, inositol, triphosphate, and calcium. And these also act through a G protein that activates a second messenger, but it's not cyclic AMP. For example, calcium can be activated through protein kinase C. And the calcium can trigger a variety of actions, including exocytosis, muscle contraction, and cytoskeleton fine management. Calmodulin is another one. This is a group of 4 protein kinases. Calcium binds to these and triggers a host of enzyme activations. And oftentimes calcium and cyclic AMP will act in concert with functional overlap and integration. G protein coupled receptors are involved in vision, smell, and taste as amplification. So for vision, rhodopsin is present in the retina. It absorbs photons and converts cis-retinal to all trans-retinal. The rhodopsin conformation changes, and G protein is converted, transducing and exchanges GTP for GDP, and then cyclic GMP is reduced, and that is the second messenger. So the prolonged stimulation of rhodopsin in vision is reversed by an inhibitory protein called arrestin, and you can also see this same mechanism for smell and taste. So when we look at this, these actions in vivo, you have vasopressin, and you have these complexes occurring, epinephrine, light, acting on these proteins like rhodopsin, with coupling to these complexes, which cause decreases of cyclic AMP or increases of cyclic AMP or decreases in these acts. So here's taste and odor and uh, uh, blood pressure, stress hormone, and vision, and you get these changes with oftentimes with ions being reduced in the system. And that will do it. Thank you.